In endocytosis, the cell's membrane surrounds a part of exterior environment and buds off as a interior vesicle. In exocytosis, an internal vesicle fuses with the plasma membrane and thereby releases its contents to the outside cell. Particles or macromolecules that are too large or polar pass through the membrane, then are taken into the cells by a process called endocytosis. Phagocytosis is one of the 3 types of endocytosis. Phagocytosis, which means cellular eating, is part of the plasma membrane. It engulfs the particle. A unicellular protists use phagocytosis for feeding, and some white blood cells use it for defending the body. Another type of endocytosis is called pinocytosis. It is a small vesicles that pinch off from the plasma membrane, bringing small dissolved substances and fluids into the cell. Pinocytosis means cellular drinking. A type of endocytosis called receptor-mediated endocytosis is used by animal cells to capture specific macromolecules from the cell's environment. For example, most mammal cells use this method to take up cholesterol. The cell uses receptors that recognize and bind to LDL particles. The receptors are clustered together in a reinforced membrane called a coated pit. The LDL particle contains 1,000 or more cholesterol molecules at its core. A monolayer of phospholipids surround the cholesterol core and is embedded with proteins called ApoB. These ApoB proteins are recognized by the receptors in the cell's membrane. The receptors in the coated pit bind to the ApoB proteins on the LDL particle. The pit is reinforced by a lattice-like network of proteins called creatinine. Additional creatine molecules then add to the lattice, which eventually pinches off a part of membrane which are attached to the LDL particles. Inside the cell, the vesicle becomes uncoated as the creatine molecule dissociates from the vesicle. Although it's not shown here, another vesicle then fuses with LDL containing vesicle, causing the pH inside to drop and force the receptors to dissociate from the LDL particle. After all of this, the receptors are released from the LDL particle and the vesicles fuse with the lysosome. A lysosome then carries the enzyme that is capable of digesting LDL particle into amino acids and fatty acids. At the same time, cholesterol is released into the cytoplasm. The cell either uses the cholesterol immediately or stores it for later. The cell's LDL receptors are returned to the plasma membrane, and once the plasma membrane, they can take more LDL particles by receptor-mediated endocytosis. Exocytosis is the process by which a variety of substances leave in the cell. For example, newly made proteins designated for secretion are packaged into vesicles inside the cell and transported to the plasma membrane. The vesicle membrane and plasma membrane fuse together, and the contents of the vesicle spill outside the cell while the vesicle membrane is smoothly incorporated into the plasma membrane.